Welcome back, Life Science learners, to our final segment for today. We've looked at types of mutations, we've looked at chromosomal mutations, we've looked at gene mutations. In gene mutations, we've looked at point and frame shift. Now we're going to try and understand what does the effect of these mutations have on the amino acid sequence. So when we talk of effects of gene mutations, we often refer to the effect that it has on the protein. So mutations may have very different and varied effects. This depends on several factors. One needs to look at the effect that it has on the protein that is produced, as well as the impact that it has on the type of protein and the nature of that protein. Often, if that protein is an important protein that's involved in important cellular processes, it means that that protein now does not carry out an important function. Sometimes we see that certain mutations may occur but not have a significant impact on the individual. When we started the segment, I mentioned that that there are mutations that occur, and I asked the question about, am I full of mutations? Do we have mutations? Yes, we do. The fact that I'm left-handed means that there has been a mutation that has altered the sequence of the gene that gives me the ability to be able to use a recessive gene, which is in my case left-handedness. In terms of the color of my skin, my hair, the ability of me to be able to roll my tongue, the ability of the hitchhiker's thumb, those are characteristics that are all as a result of variation. So what is important that we recognize is that mutations result in changes in the gene, but more than that, that is what produces variation in a population. So when you look around in your class, and if you look at your family, you will see that we all are different, yet we have the same set of parents. And that's about the effect that mutations and genetic recombination has. So let's remember that when we're looking at the effect of mutations. So thus, the effect of mutations can be categorized as follows: a neutral mutation, mutations that are harmful, sometimes lethal, and occasionally beneficial. Let's try and unpack what a neutral mutation is. The word neutral essentially refers to something that does not have an effect. So we refer to it as being harmless, or in some context, a silent mutation. Some mutations can occur in the non-coding DNA and thus do not affect the synthesis of protein. Also, several codons may code for one amino acid, and hence a mutation may have no effect on the mutated codon, which at the end still produces the same amino acid. Thus, the sequence of the amino acids in the chain is not altered, and we say that it still produces an unaltered protein. So we say that that's a neutral mutation as it has not affected the protein chain. However, we have harmful mutations, and often these would be mutations that cause genetic disorders. Most of these disorders are autosomal recessive, and I'll explain the concept what autosomal recessive is. And these can be passed down from one generation to the next. These mutations often affect an important protein molecule. An example of this would be albinism or even sickle cell anemia. So when we look at harmful mutations, we refer to these mutations as mutations that affect a specific gene. With the example of sickle cell anemia, we know that that's a mutation that alters a single amino acid. And the alteration or the change of a single amino acid affects the entire shape of the red blood cell. And so we see the impact of a single mutation impacting an entire protein sequence. We also know that albinism is a mutation due to an alteration of a gene where the pigment that produces, the pigment which is melanin, which is produced to give skin color, is lacking. And so this is the effect of what we refer to as point mutations causing a harmful effect on the individual. So if we were to look at, for example, sickle cell anemia, we see that this is the normal gene, and that normal gene which has been produced has a sequence of glutamic acid as the amino acid there. However, due to an alteration in the nucleotide sequence, a single base mutation, alters the amino acid sequence where that specific amino acid now has changed to valine. And with the effect of that, it means that there is an abnormal hemoglobin molecule that is produced, and which then in turn alters the shape of the red blood cell. When we look at the red blood cell, we can see it's got a healthy round shape, biconcave shape. However, these cells tend to be abnormally shaped and often described as sickle cell. And the problem with these cells is that they tend to clot up and stick together and do not pass through the capillaries, whereas these cells easily flow. It also means that because of the abnormal shape, the ability of these cells to transport oxygen has been compromised, and they transfer significantly less oxygen when compared to healthy, well-shaped red blood cells. We also know that there are mutations that can result in a beneficial protein. So this is when the environment in which an organism lives changes constantly and may lead to the emergence of a beneficial mutation. Mutations may cause genetic variation that could lead to natural selection. And the advantages of these are then passed on from one generation to the next. Having just looked at sickle cell anemia, and we know that that mutation can be harmful. However, in the context of the environment changing, when the environment changes and a mutation gives an individual a better chance at adapting to that environment, we say that natural selection has taken place. And so what drives the survival of the fittest on earth is the fact that some individuals are different to others based on variation. Where does that variation come from? It comes from the mutations. So mutations drive change. When that change gives an individual an advantage at surviving, they have a better chance of surviving. So with sickle cell anemia, we do know that individuals that are affected by sickle cell anemia are generally having problems in terms of the amount of oxygen being transported. However, when these individuals who live in areas that has a high prevalence of malaria, these individuals are not susceptible to being infected by the parasite that causes malaria, because the parasite that infects the host uses the red blood cell to make copies. And because of an abnormally shaped red blood cells, these individuals become naturally immune to being affected by malaria. So we see that the environment, when it changes, for example, in this case an environment which has a high amount of exposure to the parasite for malaria, these individuals in that environment have a significant advantage. And we say that they're better suited because of a mutation. And that becomes a beneficial mutation to the individual. And finally, when we look at other examples of beneficial mutations, as I mentioned, there is an example of increased bone density. So individuals in a population in America, an individual was met with an accident, and they realized that when they tested his DNA, and he walked out of an accident scene unscaled, unhurt, and this showed that he had an abnormality in his gene makeup which produced a high density of bone tissue. And so that meant that that characteristic gave him an advantage. The other example we spoke about was malaria resistance in sickle cell anemia individuals. There's also a population in Italy that have a gene which has allowed them to be able to consume large amounts of cholesterol and yet not be affected by the effect of consuming that. So the protein has changed, and that has allowed them to be able to process meat or diets high in cholesterol. So these are examples of mutations that are beneficial to the populations. So as we get into this, we need to unpack what DNA profiling is as our last segment in this lesson. DNA fingerprinting or DNA profiling is a chemical test that shows the genetic makeup of a person or other living things. It is used as evidence in courts to identify bodies, to track down blood relatives, as well as to look at cures for disease. If we were to look at an overview of DNA profiling, it is very simply a process where a DNA sample is collected, and that sample is treated with enzymes called restriction enzymes. And this allows for the DNA to be cut up into several different lengths of fragments. We then subject the DNA fragment to a treatment of electrophoresis. We know that electrophoresis is a technique that is used to separate out the DNA into fragments based on the size of their length. This is based on that DNA is negatively charged and that it would be separated due to the attraction it may have under an electric field. This is then subject to a process of electrophoresis, which is an apparatus that is used to separate out the gel. The DNA is then trapped in a gel, and that is then photographed to produce what we refer to as a DNA banding pattern. So the use of DNA fingerprint can be based on the fact that we all have a unique set of non-coding DNA. And in that non-coding DNA, we find that we are all having unique short tandem repeats. And these are short segments that constantly repeat. And that is what is used in identifying the similarities or differences between two samples of DNA that may be found at a crime scene or used to determine the relatedness between different individuals. Okay. It's important that we know that it can also be used in medicine in terms of being able to identify genetic abnormalities of individuals. And the use of DNA fingerprinting has profound number of uses in terms of determining identity, determining relatedness, and it's currently even being used to track animals that have been poached, as well as plants that have been illegally transported or moved across from one place to another. So we can use DNA profiling in the conservation of plants and animals as well. So guys, if we look at a question, and I'm drawing back to a gene which is used in the tolerance of to high cholesterol diet. So a village in Limone in the northern shores of Lake Garda in Italy has a population of about 980 inhabitants. About 40 villages have an extraordinary high level of blood cholesterol, yet upon examination their arteries have found no harmful effects. Genetic tests show that they possess a genetic mutation in which they can produce an altered protein with just one amino acid that's different to the rest. And this altered protein gives them the ability to be able to process that. This improves their ability to be able to effectively clean up excess cholesterol. And this means that no matter how much of excess cholesterol they consume in their diet, that is always collected and disposed of from the blood. From the village records, it is discovered that all these carriers were related and descendants from one couple who arrived in this village in 1636. Generally, the people of Limone live longer and show a greater resistance to heart diseases. So this was a question that's based on the effect of a mutation. Explain what is meant by the term mutation. So guys, again, a mutation refers to a change in the sequence of a nucleotide. That's essentially what the context of today's lesson was. We've got to identify the type of mutation that was produced, and in this case, the mutation that was produced was a beneficial mutation, and the reason that happened was that it produced an altered protein that gave them the ability to be able to process extra cholesterol in their blood capillaries. And so that is seen as a significantly advantageous mutation. So guys, we've rushed through the segment, we've tried to look at DNA profiling, we've looked at the effects of mutations, and we've recognized that mutations can be beneficial, harmful, lethal, and even neutral. It's important that we understand that mutations drive variation in the population, and when we move on to evolution, we often refer to natural selection. And that natural selection is driven by the variation in the population. So to sum it up, mutations are important in a population because they create variation, and that variation is important to the species being able to adapt. So we've wrapped this segment up by looking at mutations. We've looked at the effect of mutations, and I hope that you guys go back and reflect on why mutations are important in a population and how they drive natural selection. Thank you for staying tuned. Wish you well, keep well, go safe.